Welcome to the public lecture by the Faculty of Science at the University of Johannesburg in partnership with the UJ Library. My name is Fidel Tugizimana from the Department of Biochemistry here at UJ. So I will be guiding you through this public lecture. We start with an introduction of our speaker, Professor Justin van der Hooft, followed by the presentation. Then we conclude the public lecture with a question and answer session. So if you have questions during or after the speaker's presentation, please post them in the QA section. Professor van der Hooft obtained his MSc in molecular science in the Netherlands and took up a PhD position in analytical biochemistry, where he developed methods for systematic metabolites identification and annotation with both mass spectrometry and NMR techniques. In 2012, he obtained his PhD and moved to Glasgow in UK, where he worked on several postdoc projects. There, he set up a collaboration with Dr. Simon Rogers and the concept of MS2LDA to discover substructure in tandem mass spectrometry data was born. In 2017, Professor van der Hooft moved back to the Netherlands to start on a joint postdoc between Prof. Peter Dorsen from US and Dr. Maxime Medema in the Netherlands. In May 2018, he started on a 3-year grant from the Netherlands eScience Center that further expands the work on metabolome mining. His research vision is to close the gap between what we can see in metabolomics and what we can learn from it. He focuses on developing computational metabolomics approaches inspired by two other fields such as of natural language processing and genomics. In 2022, this year, a new eScience grant is starting that will further build on previous work on NP linker as a framework to perform systematic integrative omics mining and the paired omics data platform that records paired omics datasets, as well as validated gene cluster mass spectral links. He is using plant material, microbiome, and human food metabolome as a prime applications, and understanding the metabolite complexity of these metabolite mixtures, those that are known, those are not yet known, can boost our insight in molecular mechanisms underpinning the regulation of growth, development, and health. In today's presentation, Professor van der Hooft, he will be sharing with us the tools that he has developed with collaborators that are 4IR inspired computational strategies to mine and interpret metabolomics data. I will now hand it over to our speaker, Professor Justin van der Hooft.
Thank you so much for your kind introduction, Dr. Fidel, and also for the kind of invitation to do this public lecture, Fidel, and also Professor Liz. Thank you so much. Let me share my screen. And then we can start. So my name is Justin van der Hooft, and I'm currently an assistant professor in the bioinformatics group at the Wageningen University. And my computational metabolomics group is now consists of 6 people. And then we also have a number of master students and a bachelor student working with us. So there's 3 PhD students, David Meijer, Nick de Jonge, and Kevin Mildau. And there's a postdoc, Mitya Seduk, and a research assistant, Joris Lauwen. So if you want to know more about our group and what we're doing, there's a brand new website. The URL is provided on the slide. So I welcome you to have a look there and browse the various projects and people and what is going on. And also we will post regular news updates when there is something to announce. So as Fidel already kindly mentioned, is that most of the tools and developments in metabolomics are really teamwork, right? It's a multidisciplinary science, and as such, you see that a lot of the tools are made by different people because each can contribute their own expertise and background knowledge. And here are the various teams that I was part of over the last years. So Fidel mentioned them, in Wageningen, in the Dorstein lab, in San Diego, but also I've spent a number of years in Glasgow Polyomics in Glasgow, in Scotland, and you can see and actually you could find me on most of the pictures of this group picture. And I also want to do a shout out to the eScience Center that has been currently funding the iOmica project, from which I show a number of results, and also the new grant, open eScience grant, will kick in soon, and then we will start to work further on combining genome and metabolome mining tools. So most of my work, and also I think many of your work, usually starts with simple questions like, we have these two molecules, who's producing them, which microbe, for example, and what is their role, what is their function in the ecosystem? Do they make the plants happy or sad, or do they make us happy or sad when we eat the plant that contains these molecules? Sorry. However, it seems to be a simple question, but it's actually not so simple as it is, right? Because we don't look at molecules, we typically see spectra. So today, my talk will be mainly focusing on mass spectrometry-based metabolomics. I will get back to that. But so we typically see this mass spectra, and then the first question is, what is in there? And then we can answer this, start to answer the question, what is it doing? So in my work, I get a lot of inspiration from nature because I study, after all, natural extracts, but also humans have been inspired by nature and natural patterns for a long time. So, for example, the inside of this church, I took this picture because already in the 11th century after Christ, when they started to build this church, they designed it with natural patterns in their heads. You can see the flowers on the right hand side, for example. And also my work is kind of trying to find the patterns that are inherently there in nature and exploiting them to increase our understanding of spectral data from natural extracts. So I know that in Johannesburg there's quite some work being done on plants and microbes and how we can use them to help us to solve problems like drought stress, salt stress. These are really issues, I think, also in a huge part fueled by the climate crisis that we are facing and that we're actually already in, that we will need to tackle in order to maintain a good yield of our crop and staple food in order to keep up with the demand of food of the world population, especially when we realize that there should be a more plant-based transition to also reduce the impact of the on the climate from our food. So, and having said all that, it is also true that we also still need to look at how nutritionally diverse our food is, and also what is the impact of all these microbial metabolites that we more and more are starting to decipher. So basically, in summary, a lot of these small molecules, even though they're very small, they can have a huge impact on growth of plants, of microbes, on us, and also on our health and the health of the complete ecosystem. So the challenge is that we now know that bacteria, fungi, and plants, they produce a large and very diverse arsenal of high-value molecules. Here are some examples on the slide. So I'm not going to go over them one by one, but the bottom line is that these molecules were found in nature, isolated, structurally identified, and then actually we as humans found a way to exploit them and maybe not where they originally were meant for, but we found a way to use them to our benefit. I think in a lot of sense, the antibiotic is a good example of a molecule that also has the same goal in the ecosystem, right? Killing neighboring, competing bacteria, right? And now we use them also to do that for us, to help us to kill and fight off pathogens. So the real challenge is to couple the structural information of molecules to the data, right? And especially large molecules, and that's already everything larger than lovastatin on this slide. I consider as a large small molecule, and there the possibilities of how the atoms are placed are starting to become endless, and therefore it's really hard to do structural annotations. So we need innovative tools, because the standard way of doing things is like we select things, we run the mass spectrometer, we buy standards, and we check. And that works, but only for a small amount of molecules. So the state of the art is typically that you get up to on average 5% of the features in your data annotated. And that typically also with the most well-known compound classes. So that also means that on average 95% are still unknown. Unknown source, are they from the plant, are they from the microbe, are they from the environment, and unknown pathways. However, and then think back about what I said previously, in natural mixtures, most metabolites are connected somehow, right? Through pathways, through substructures, and therefore I want to use that to close the gap between what we can see in metabolomics, the spectra, and what we can learn from it, the structures, the biochemistry, the functionalities. So I designed a research agenda that is composed of 3 major pillars. So we will see examples of that today. So one is based on machine learning and networking tools to read the metabolite structure from the spectra. One is to can we find ways to prioritize chemistry in complex mixtures? And one is to link the metabolomics profiles to other omics profiles like genomic profiles, genome mining results, to gain functional insights. And I thought it was nice to share that untargeted metabolomics in general seems to be really on the rise. And I can demonstrate that if you type in this in PubMed, you see this kind of graph. With last year, I think nearly 1,000 publications that mentioned the term untargeted metabolomics, and there are variants there which I did not cover now. So I mean, and you can clearly see that this is a growth, right? And I think it's because information got more sensitive and faster. We have more generic extraction protocols available to us, and also it became higher throughput, and we have these fractionation robots that can help us in some labs. But I think one of the main drivers of all of this is the support by the fourth industrial revolution, the 4IR, that is really also kind of pivoted in Johannesburg, right? Computational metabolomics tools. So you can see here how that is also growing. Virtually non-existing before 2000, the term in literature, and now you see it's on a steady rise as well. And the role of 4AR in metabolomics or computational metabolomics is really to go from the spectral data to interpretation, right? So we can use networking tools and we can use substructure-based tools to create informative networks to then try to answer the questions on like what kind of pathways are involved in abiotic stresses, as you can see from this graphical abstract from work by Morena Tinte and also with Fidel that came out last year. And if you then zoom in a bit on the role of 4AR, then you can see that it can play roles in many aspects of the metabolomics pipeline, not only at the analysis part that I will highlight today. Yeah. So if you are interested in that, please have a look at the paper, and you will see that it really can impact many aspects of the whole pipeline. So I know that you have a various background, right? So and because it's important to kind of get some of the concepts like straight in our minds to continue the talk, I want to show you briefly what is a mass spectrum, at least what I understand for the talk, right? So we have various masses coming from the molecules that are in your extract, and typically in a mass spectrometer they are somehow concentrated and then measured, detected, yeah, and then they are separated based on the mass over charge ratio. Small molecules on the left-hand side, the larger ones on the right-hand side, and we can also kind of get a relative imbalance. And then you get your mass spectrum, right? However, for today, we will actually focus on one step further, where we actually isolate one of those peaks that we just obtained. So hopefully one type of molecules, and then we add energy in the form of an inert gas in a controlled manner in the mass spectrometer, and then we get a so-called MS2 spectrum or MS-MS spectrum or tandem mass spectrum. You can find these terms in literature interchangeably, in my opinion. Yeah. So, but they all mean they all point to this kind of fragmentation spectrum, right? So we don't no longer see the complete molecule, but parts of it, and we can use that to find patterns. Yeah. And this picture I took near a beach near San Diego when I visited the Doorstand lab. And I took this picture for one reason, right? Because within a few seconds, my mind saw two groups. And humans are really good in finding these patterns when it comes to this kind of animals, right? And but you can imagine that if you do this for spectra, we can also try to group them based on spectral similarity into families, but we can also try to extract the building blocks of metabolomics, substructures. Here, it would be the building blocks of the animals, let's say the heads, the feathers, the etc., etc. In our case, it will be the substructures of the chemicals. The problem is that we are good in animal grouping, we're not so good in spectral grouping with our eyes. So there we need this computational support, right, from the computational metabolomics tools. And we can then use this grouping to propagate structural features and to annotate families as a whole. So there's a lot of literature out there and already reviews and documentation on GPS molecular networking. So I don't spend a lot of time on that today in the lecture, but this slide is kind of showing you the basic principle behind it. So it all starts with a bunch of molecules. You obtain the spectra. You then cluster the spectra, the very similar spectra. They become nodes for your network. And then in the final step, you create a network by comparing the spectra in each node to each other node. Yeah. So basically, you assess how similar are the spectra, and then if they are above a threshold that you put, you draw a line, right? And that becomes your edge. So and then, by simply repeating that process, you create a network as large as the data is. So and when multiple nodes are connected by edges, we typically call that a molecular family. So what I want to spend a bit more time on today is actually the MS2LDA, unsupervised substructure discovery, because that was what Simon Rogers and Joe Wandy in Glasgow, together with me, have been developing and proposed and introduced. So there we go from text to molecules, and the way it works is that there were already back then, and even now, even more machine learning based algorithms developed for text mining. And one of them, latent dirichlet allocation, LDA, was coined to find topics, groups of words that describe something in text. Okay, you see examples on the left-hand side. Yeah, the red words, football-related words, blue words, business-related words, green words, environmental-related words. And the algorithm, when presented with a lot of data, documents, is able to find out which words are often co-occurring within and across documents. Okay, so let us think a bit about that. So, and let us look at what's happening. So in one document, we can identify one or more topics. In this case, you see also two groups of words. And also, they can be the same across documents. So that the blue words topic is present in document 1 and 2. And when we think about substructures, that's exactly what we want, right? So one molecule can be composed of multiple substructures, but also the same substructure can be present across multiple molecules. So therefore, this approach seems to be very suitable to also try out on metabolomics data. Yeah, where the words become the fragments and the documents become the whole spectra. And that's what we did. Yeah, so we gave it a try, and yeah, so because it is already some time ago, I can tell you that, of course, after some trial and error and optimization, it seemed to be working quite well. Yeah, so because MS2A substructure discovery was able to find a range of very biochemical relevant substructures in this case beer extract. It's a bit early here to talk about beer, maybe, but you know, maybe a good appetizer for the evening. So we took some Scottish ales, and there I was able to find this range already here presented on the slide of amino acid related, sugar related, and aromatic substructures. We also validated with standards, yeah, because back then there were 5,000 GPS standard spectra available and 2,000 mass spec, and we took them and we also validated it. So I chose this example for two reasons, and the first one is that the first ever MS2A result that I checked and I was able to say, okay, yes, there is promise, was because I saw this motive for adenine appearing, and also here we look at the same or similar motive where we see that these three structures, they share the same adenine core substructure. And you can see on the left-hand side that the red peaks are associated to the motive, to this pattern in the data. But you can also see that this pattern it looks similar, but it's certainly not the same. So that's good to know that this probability-based machine learning approaches, some variation in the data is okay. It can cope with that. And also, we can actually see how these multiple substructures work, because we also see the green loss, and that is devoted to the ribose-related substructure in adenosine, and the whole molecule of adenosine in the middle is actually built up from two substructures that we could see. So I'm I'm also looking at the time, so I will try to speed up a little bit now. So there's also a database, MotiveDB, that we coined to collect omics detectable substructures. Yeah, and the way it works is that if you annotate substructures in your data, because that's maybe something good to also mention, the LDA algorithm doesn't annotate the stuff for you. You still have to do it yourself. So that's what I've been doing during my postdoc time, and I'm still doing now and then, often in collaboration, is to identify new master motive substructures. To then, in this case, I identified this one, annotated this one, sorry, with the lactone ring of actinomycin D, and then now it is part of an annotated motive in a motive set for Streptomyces and Selenospora. So next time somebody comes with bacterial data related to the strains, they can use these annotated motifs, and they no longer need to re-annotate the the actinomycin-related molecules, because this motif will find them for them. So I promised in my abstract some applications on plants. So this is the case study that I will present a bit from. So it's from the Rhamnaceae plant family. It's growing worldwide. But what's important to remember now is that we have two clades, the rhamnoid clade and the ziziphioid clade. And what's also good to realize is that we have more and more computational support, and it's really nice to see how the labs from Dave Wishart and Peter Dorstein and others have been cherishing this chemical compound class ontologies. So ways to automatically categorize molecules based on their structures in a computational way, and these ontologies are really organized in a hierarchical manner, akin to the tree of life with the kingdom, superclass, class, and subclass. And what is important to notice is that these chemical classes are also related to each other. So you can see how you go from lipids and lipid-like molecules all the way down to the level of triterpenoids or terpene glycosides. Okay, so that is something good to keep in your mind. Yeah, that we increasingly get the tools that help us to automatically categorize molecules, and we can use that because when we do a molecular network of this Rhamnaceae data obtained in negative ionization mode by Kyobin Kang, who is now having his own group in Korea. So he obtained the data, and together with him and Madeleine Ernst, we have been analyzing the data. And you can see how in the molecular network, when we classify the molecules based on the Molden enhancer workflow, you can see all plant-based classifications. So flavonoids, phenolic glycosides, and triterpenoids, which make a lot of sense, right? So that was good to know also as a kind of validation for the whole computational workflow. This is all published 3 years ago now, so if you're interested, have a look at the paper. Some examples, more examples from that paper and follow-up work is that we could also dig into this molecular families. I explained what that was, so the nodes connected based on spectral similarity. But now we also map the substructure presence absence on top of them, and then you can start to see how one molecular family is actually breaking up in 2 subfamilies, one based on the camphoral substructure, which is an flavonoid often occurring in plants, and the other one on based on quercetin. And in a similar fashion, we could look at the triterpenoid family. Again, a lot of nodes with a lot of edges, but now we can start to really pinpoint where particular benzoic acid conjugates are occurring based on the color here. Yeah. So again, it's kind of a breakup in subfamilies. And we can also now zoom out and use all this information to study the clade chemistry, right? Remember, I told you there was 2 main clades, rhamnoid, ziziphioid, and we can see how some chemical ontologies are more occurring or more present in one clade than the other. So actually, it helps us to identify particular clade chemistries. And I highlighted here, it's a bit, maybe a little bit, how you say, biased now, but it helps you to look where to look for. And I think some patterns are really strong, like for the flavonoid glycosides and also for the triterpenoids. So I want to do a shout out to Madeleine Ernst, who has been championing the Molden enhancer work. And it's a workflow used worldwide now, has been cited more than 100 times, has been applied to many different types of extracts now, and it really helps you to bring together a number of the computational metabolome mining workflows that has been coined over the last years. Also, it has been used by people in the group of Dr. Fidel. So if you are interested, I'm sure that he will be happy to point you to the people there, and they can help you also with applying it to your own data if you would be interested into that. So I also wanted to introduce this concept of mass spectral embeddings, and it's actually a bit similar to the MS2LDA world, where we make the analogy to text mining, but now we no longer look for topics, but we really look for similarity in text, in sentences, and in spectra. So in other words, can we find out using machine learning, there are algorithms that can find out that likes and loves mean roughly the same thing, and cake and cookie are also similar, not the same but similar, etc., etc. And now the idea was, can we use that also to do that for fragments? So again, a word is then a fragment, and the sentence becomes the whole spectrum. And then we want to be able to assess if a fragment, even though it's not exactly the same, like A prime, but almost the same, can we find a way to establish that relationship? And for that, together with Florian Huber and Simon Rogers and others, we transformed existing word2vec into spec2vec. So word2vec takes in words. You can see examples here. So likes and cake from the last slide, and you can see two embeddings here, 2 dimensions. Sorry. So one I called positive affection, and one I called hot drinks. And then I gave numbers to the words. So which word would fit more into one of those dimensions, right? And well, you can argue about it, but we won't do that today. I will just show you that if you would, you can also learn this from a lot of text documents, and then you would maybe get numbers like I present on the slide now. So, and then we have simply 2 dimensions, and then of course it's up to us to do something with that, or to interpret that, or to use that in follow-up analysis. In our case, we get many more dimensions, 300. And we have many more fragments and losses that we throw in, and then we use all the mass spec data to also, find out the relations between fragments and losses in the data. And the idea is then that we can use that also to, to compare spectra, because if we know the relation of one fragment, we can also add up all the fragments and losses in the spectrum. And we end up somewhere in this 3-dimensional space. And we can do the same thing for another spectrum. And then we can also find out how far apart are they in this 3-dimensional space. And that's exactly what we did. So we use that to design a new way of mass spectral similarity scoring. And then we compare that to existing ways of doing this similarity scoring, for example, for the task of library matching. And there we could see that, and this plot shows that, is that the accuracy, so the percentage of correct hits, is higher across the whole board than for the classical way of doing it. And also you can see that even at getting back 50% of spectra, so for each 10 spectra you send, you get 5 back, so that is a pretty decent retrieval. You still have a pretty high accuracy. So the bottom line is that the mass fragment correspondence, if you only use that, it's not sufficient apparently to get the full information out of your data. And therefore, I think spec2vec and other machine learning based approaches really have a lot of promise when we can further optimize them. And also, I want to briefly highlight that this kind of approach seems to work especially well for larger molecules, or larger small molecules. And that's exactly the range where we still have most problems, right? Because tools like Canopus and Sirius and Cosmic, they're brilliant, but they're especially brilliant for the smaller small molecules, so up to 400-500 Dalton, and above that we still struggle nowadays, and I think this kind of developments will really help to also attack that area of unknowns. And then if you're interested, there is this case study on complementary similarity metrics in a recent review by Mehdi Benadir, Cuban Kang, and others, including myself. I will not talk about that in detail today if you have time, but the bottom line is that if you use different of these mass spectrometry metrics like modified Cosin score and spec2vex score, then your network will change. Yeah? So, and of course, what is then the best choice? Well, that depends on your question and your data, I'm afraid. Huh? So this networking there's no gold standard. So yeah, so my main message to you would be to really report well what you did and which choices you made and parameters you used. In that way, people can always repeat it and modify it and then redo the analysis if they want. So I want to do a brief shout out for my PhD student Liek de Jonge and Joris Lauwe and Florian Huber. We are working on MS2 Query, which is a machine learning based framework for library matching and analog search. And it uses spec2vex that I just explained, and MS2 Deep Score, another mass spectral embedding based approach, and other scores, and it combines them. And it can do really fast analog search, which is a real benefit compared to existing approaches. It will be it will help you to easily implement what I just told you into your metabolomics annotation pipeline. So yeah, unpublished still, but keep an eye out, because we hope to change that this year, right? Go towards at least posting a preprint. So I want to shift gears a little bit in the final part of my seminar to the linking omics research pillar. If you remember from my start of the seminar, right? The 3 pillars. So the one on the right was about linking omics profiles, and that's what we're going to do now, or at least briefly discuss. So it would be ideal if we know from each molecule in our extract where it is coming from, which bacteria is producing it, because it can help us to also find out possible functions, right? And the strategy that is now being more and more developed is to sequence the genomes of the bacteria, mine for gene clusters in there, and then link it to the structural information. And in that way, we can make this circle, right? So we can link the molecule to the producer and also obtain additional structural information from the genome. For example, about stereochemistry of amino acids. This is really hard to get from the mass spectrometer. And in the iOmica project funded by the eScience Center, we've been working on various solutions at the data level, the structure level, and the substructure level. But at the data level, there's now the paired omics data platform. And what it does, it brings together, sequence genomes, for example, from NCBI or GTI, with metabolomics data, for example, GMPS massive or metabolites. And it records the links between the same samples. And that is really useful because we can use that to mine for novel links. But not only that, you can also record known or validated links between gene clusters and gene cluster families and their products. So, and that helps people like myself and others to develop novel tools to link gene clusters to molecules and mass spectra. So we designed quite a straightforward way of adding your data to the platform. So if you have mass spectrometry data of sequenced genomes, I highly encourage you to contribute. And or if you have sequenced genomes and some room for mass spectrometry, then why not do the LCMS and add that to massive and then upload it to the records to the paired omics data platform. So yeah, there's various sections to go through, and at the end you can make the actual links between the LCMS files that you point to the right URL, and the different labels for the genome and for the minimal metadata on the methods that were used, like the sample growth and the extraction and the instrumentation methods. And that is included to make sure that you can make proper reuse of the data, right? For example, if you are only interested in peptides, then you want to make sure that they are included in the measurements, yeah? and that the right mass window was used for obtaining the mass spectrometry data. So when we zoom out, it is part of a already quite complex genome mining and metabolome mining ecosystem on itself, right? So this is a very complicated picture. I do apologize, but that's also because it's quite a complex collaboration of tools that in the end lead to predicted matches between the genome and the metabolome. Yeah. So you can see the paired omics data platform is somewhere in the middle as a kind of a start or initiator, and you can then see the whole kind of genome mining workflow on the left-hand side with anti-smash developed by Professor Marnix Medema and colleagues, and on the right-hand side the metabolome mining workflows, for example, based on GPS from the Peter Dorsen lab. We also see tools to cluster and group gene clusters like BigScape and BigSlice, again spearheaded by Dr. Marnix Medema, and you know, the molecular networking we discussed, and MS2LDA we discussed. And then we can try to bring that together, and then the first examples of platforms to do that in a systematic way are MP-Linker and MP-Omics. So I believe the future of this network products discovery will be in a multi-omics way. When we more and more have this multi-omics data available, we can create gene cluster families, we can create metabolite families, and we can then do this so-called strain correlation-based matching, where we check, okay, which strains are these genes of this family found? In which strains are the spectra of this molecular family found? And do they match up or not? And then at the second level, we can do feature-based matching, right? Does this gene cluster produce a certain substructure? Oh, does this molecule has the same substructure predicted in it based on its spectra? And then we can see if it matches or not. And we can use complementary scores to filter and rank the most plausible links between the gene clusters and the spectra in our data in order to find out the links between the molecules in the end and elucidate the molecules based on the genetic and the metabolic information. So here is also a call to action. Sharing is caring. So we have data, mass spectra, gene clusters, structures, metadata, paired data that we already all discussed or heard about. And we have this set we have this, and this is not a complete list, but this is just an example, right, of a number of repositories and databases that basically are dependent on us as a community to contribute, right? If we as a community contribute, then our knowledge, our complete knowledge grows, and tools will also improve. Like if we talk about SpecVec, it's trained on public data, like from GMPS, right? So we are the tool, the more spectra are annotated spectra are being submitted to GMPS, the better the tool will perform. So, and in a similar way, MyBig now contains slightly more than 2,000 validated gene clusters. But if you have them, and if you publish a gene cluster, then why not also submit it to MyBig so that in the next iteration of a tool, this knowledge can be used and reused. So that is my call to action. Sharing is caring. And if we talk about outlooks and some inspiration, I would love to study ways to use this mass spectral embeddings that we discussed also to do more annotations, for example, chemical classifications, like we can see here. Here each dot is a spectrum and each color is a spectrum from the same molecule. So you can already see that somehow they are usually grouped quite nicely together. But when we zoom out a bit and we only look at the chemical compound classes, then we can see that actually we can start to see patterns in there as well, based on that on the right hand side we mainly see the flavonoid glycosides, on the left hand side we mainly see the O-methylated flavonoids, etc., etc. So zooming out again, we can I predict that in 5 to 10 years, we will have the infrastructure, so in the collaboration with many groups around the world to do this large-scale substructure-based analysis, where you feed in your metabolite mixtures, metabolomics data, and you can do class annotation, substructure annotations, and also this networking analysis in order to really fuel and propel large-scale metabolomics analysis. And if you want to know more about this, I can recommend to read the review, but I'm biased because I'm one of the authors. But I think it gives you a really nice overview of where we are and where we could go, both for mass spectrometry and NMR-based analysis, I have to say. So I'm going to leave you with some inspiration and some, what else can you do? So this mass spectral embeddings, I told you we had 300 dimensions, right? And I already showed you a two-dimensional plot of it 2 slides before, right? But you can also reduce this 300 dimensions to 3 dimensions. And then, and that's what we did in collaboration with the Wonder team in Wageningen, and they developed this kind of tool to kind of browse through the mass spectral embedding in 3D. And what is nice is that you can then see the 3D shaped clusters, and also you can hover over the dots, the molecules, the spectra, and find information about them. So in this case, each color is a compound class, so you can already see from the colors that indeed same classes are often grouped together, which is encouraging because it means that the mass spectral embedding actually learned some chemistry. So yeah, on that note, I want to really thank you for your attention. And I've showed you solutions for based on the data level, the structure level, and the substructure level. And if you're interested in all of this, feel free to follow me on Twitter and or my group, and the handles are on the slide. So thank you so much.
Thank you, Justin. Thank you for the wonderful presentation. It's complex but very informative, and we can really see that the metabolomics annotation of the metabolites with the aid of computational tools, it's really providing solutions that have been there for a while. Annotation of the metabolites is really a bottleneck, a challenge in extracting information, biological information from metabolomics data. But from this presentation, you really showcase that with the development of these tools, it will indeed help a lot in gaining more insight from the metabolomics data that we acquire. So we go now for Q&A session. There are a few questions that I'm checking on time. We have almost 10 minutes left, but maybe you can answer 1 or 2 questions. One question, as you show different tools that are being developed to mine substructures, to understand, to deduce or to elucidate structures of the molecule. To what extent are these computational tools or algorithms accurate in differentiating isomers?
That's a very that's a good question, quite detailed, but yeah. So isomers are, for those of you that do not know the term, are molecules that share the same elemental formula, but they have a different structure. So and some isomers are really different because they are not of the same compound class even, but other isomers can be very similar, right? They for example, a flavonoid molecule with a sugar that is attached to different hydrogen groups, OH groups, hydroxyl groups. That was the word I was looking for. So it depends on what kind of isomer we're talking about. So if it's really a different compound class, then I think that in many cases the chemical compound class prediction offered by Canopus or Molden Enhancer will already differentiate that. Yeah. If we talk about positional isomers of the sugar, then it will be trickier. Yeah. So you need some own expert knowledge in order to recognize the fragmentation patterns, or you use reduction time information. And in that sense, if you're interested in that, you can, for example, do peak picking using XCMS or MZMine based approaches that identify LCMS peaks. So combination of retention time and masses. And then they will be able to differentiate between the isomers if you use the correct chromatography. And then you could use that as input for feature-based molecular networking, where these isomers are then still separated. But for their structural elucidation, of course, it will not always be possible to do it fully on MS only, and you may also need to rely on NMR or other tools to get complementary information. Yeah? My final word on this is that if you talk about stereo isomers, so the difference between D and L amino acid in a molecule, that is very hard for mass spectrometry. But there we can use, for example, complementary information from the genome, if available, or other analytical tools like CD, circular decorism, and others to differentiate between the stereo isomers.
Yes, thank you, Justin. Another quick question, practical questions. In the GNPS platform, is there an ideal parameters to set when you are working with plants or bacteria, or one has to trial and error type of approach?
Yeah, so that is, I think, it's a very good question. Yeah, so and the very short answer is there's no ideal setting, right? Because so of course the sample type is important, right, for annotation. But I think the instrument type and the way you obtain the spectra are even more important for the GPS settings. After all, GPS doesn't really care the type of data or the metadata behind it, right? Could be urine, could be bacterial, could be plant, could be organic matter. I mean, as long as there are spectra and precursor masses, it will do its job. But if you come with ion trap data or orbit trap data, so low or high resolution, that will matter a lot to what type of settings you should use, I think. So there is a Nature Protocols paper from 2019 by Alega Agon et al. That already details some basic settings for different platforms. So maybe that is a good way of starting, right? Identify your platform, identify those settings, and then take it from there, right? Yes, it will involve some trial and error, but yeah, that is true for all tools that do not, for which there's no ground truth. There's no perfect network, right? So.
Thank you, Justin. Another question. You showed the integration of different omics, specifically genome data and metabolomics data. How about proteomics data?
Yeah, good question. So actually, as a matter of fact, if you would go to the paired omics data platform, you will see that it is possible now to add proteomics data of the same sample. Because of course it could help you to identify active gene clusters and to as a guide to find out which are the ones that are really expressed. So what I have to point out is that the way that we implemented it now is that you need to have metabolomics data in order to connect it to the genes and or the So to the genes and to the proteomics data if you have it. So yes, we started to think about it, but yeah, I have to say there's also not a lot of studies yet where all this information is available, because you have to also keep in mind that when you do metabolomics and proteomics, that for both of them the situation, the when you do it, how you do it affects the profile, right? Whereas for the genes, the genome is much more stable. So this is also something to keep in mind. Huh? You cannot do metabolomics on one day in the sunshine and then do the proteomics a day later when it rains. Yeah, for example.
Thank you. Let me check something here. So we have almost 4 minutes left. One last question, and before we conclude, just a quick one. These metabolites in a cellular environment, they are in a different compartment, cellular compartment. Are you thinking, the computational metabolomic scientist, thinking in terms of maybe developing tools, algorithms that can, as you annotate, maybe add also that type of spatial information, cellular spatial information to the metabolites, which...
Yeah, so that's a very good question, and actually has been part of a grant that was submitted recently or last year. Yes, I would love to work toward a way to store that kind of information together with, for example, substructure patterns, right? So, for example, you identify a substructure, you can also store information on where it was found and what is it doing or which pathway is it part of. Yeah. A good idea.
Okay, thank you, Justin. This was really a wonderful, informative seminar. And on this note, if you have few final concluding words. And
yeah, sure. So I thank you so much, Fidel and Liz, for this opportunity, and I really hope that you enjoyed the seminar. So don't worry if you don't understand everything, because that was not my intention. It was also mainly my intention to inspire you and make sure that you're aware that all these developments are ongoing. And yeah, there were a lot of links out to the resources, and that will be a good start to find your resource that you are interested in to find out and to develop. And as I said, I think Fidel and his group are already starting to use many of these tools, so they will also be an excellent point of getting some information from locals if you are in Johannesburg or nearby. So thank you so much again. Bedankt. I don't know if that's also Afrikaans, but yeah.
Okay, thank you, Justin, and thank you to those who attended. And there is a survey link that has been posted. Please complete it for improving this public lecture. Thank you so much.
Bye-bye.
Bye, Justin.
The University of Johannesburg, the future reimagined.